In this video, we are going to talk about disaccharides. Recall that disaccharides is when two monosaccharides are joined together. We're going to look at the formation of the bond that will join the two monosaccharides together, and then we're going to look at its hydrolysis. At the end of the video, we are going to look a little more deeply into the chemistry behind lactose intolerance and artificial sweeteners. Okay, so formation of disaccharides is going to be a dehydration reaction. Recall that dehydration means that we are losing water, H2O. So again, disaccharides are composed of two monosaccharides. We learned previously the term hemiacetal, and now I'm going to introduce the other term acetal. So the hemiacetal of one monosaccharide is going to mean the same thing as the anomeric carbon. So the anomeric carbon in one carbohydrate is also the hemiacetal in this monosaccharide. So in this case, this hemiacetal is the anomeric carbon. Anomeric carbon. All right. Now what's going to happen is that anomeric carbon or that hemiacetal is going to react with a hydroxyl group of a second monosaccharide. So recall that monosaccharides are going to have a lot of hydroxyl groups on all of their carbons. And so what this means is that one of those hydroxyl groups can interact or can react with the hemiacetal of another monosaccharide. When that happens, this OH group and this H leave and they come together to form water, so that's H2O. And now this oxygen here can go ahead and form a bond with that anomeric carbon. And that is what is being shown here on the right. This oxygen here, let's do this in blue. This oxygen here is the oxygen that is coming from one of the monosaccharides. And then this carbon here is your anomeric carbon in the other. So this is how you carry out a dehydration reaction with two monosaccharides, where the OH group on the anomeric carbon leaves, one of the hydrogens in one of the hydroxyl groups in your other carbohydrate is removed, and then the oxygen that is left behind is the one that forms the bond with that anomeric carbon. So this new group that is being made here, this is what we call an acetal functional group. An acetal functional group is defined as a carbon that's bonded to an oxygen, that's bonded to another R group, and then it's bonded to another oxygen that's also bonded to an R group. This is how it's different from a hemiacetal, because in a hemiacetal, one of those oxygen groups that are bonded to that carbon have a hydrogen. Okay, so they'll have a hydroxyl group. So the difference is a hemiacetal has a hydroxyl group bonded to that carbon, while in an acetal, you don't have an OH, you're going to have an OR group. Okay, so now let's look at specifying the orientation of these bonds. Recall that when we talked about monosaccharides, we could classify them as alpha or beta anomers. And here we're going to have to continue to define those when we are joining these monosaccharides together to form the disaccharides. And this is important because this will make the difference between whether we have carbohydrates that we can digest versus not. And we will look at that in a separate video. But right now, we just need to be able to identify these glycosidic bonds, which are the bonds that join two monosaccharides together, and be able to name them. Okay, so the glycosidic bond is what joins two monosaccharides, and it can be oriented down, and we're going to specify that with an alpha, or it can be oriented up, and we're going to specify that with beta. Additionally, numbers are going to be used to designate which ring atoms are the ones that are joining the disaccharides together. Because recall, you do have OH groups on most of the carbons in your carbohydrates. So this means that any one of those OH groups can interact or can react with the anomeric carbon. And so we're going to need to specify it. So here's an example of such a case where we have our two monosaccharides. Here's the first monosaccharide, here's the other monosaccharide. And then we're going to need to locate the glycosidic bond. Again, the glycosidic bond is going to be the bond that comes off your anomeric carbon. So the first thing you need to do is locate your first monosaccharide and locate the anomeric carbon, which is this one here. I know this is the anomeric carbon because one, it is to the right of this oxygen in my ring. This is the standard that we use to draw our structures. So this has to be the anomeric carbon. And then I see that this oxygen is bonded to that anomeric carbon. And that is what joins then the second monosaccharide. So the glycosidic bond is this one here between the anomeric carbon and the oxygen of the second carbohydrate. or the second monosaccharide. Okay, so that means that this bond is currently being drawn below the ring or down. And that's why this is going to be an alpha glycosidic bond. Sometimes you'll see it written as a glycoside, as a glycosidic linkage, and other times as a glycosidic bond. All right, so that's going to be down. So then that means that when the bond is pointing up, again, this is my anomeric carbon. I know this because there's an OR group in that direction, an OR group in the other. And that means this is the bond that is going to be my glycosidic bond. And because it's pointing up, meaning it's above the monosaccharide ring, this is going to be a beta, just like we did when we were looking at just monosaccharides and identifying alpha and beta anomers. Okay, so that is the first part in identifying what kind of bond you have. You have to determine whether it's alpha pointing down or beta pointing up. And then you have to specify which carbons are the ones that are participating in this bond. So here in this first example, remember that we number the rings as 1, 2, 3, 4, 5, and usually there's a CH2OH group here. And then we're going to number this the same way, 1, 2, 3, 4, 5, and then there's a CH2OH group here. Okay. So this means that the first carbon in one carbohydrate, so carbon number 1, is bonded to carbon number 4 in the second monosaccharide. So that is why this linkage gets a 1,4 alpha glycosidic linkage name. If we look at the second one, again, it's the same idea. These are my carbons. These are my carbons. So we also have a 1-4 bonding, meaning the first carbon in one monosaccharide is bonded to the fourth carbon in the second monosaccharide. Okay? But it's going to get the beta designation because this oxygen here for this bond is pointing up. Let's go ahead and apply that to get a better understanding of how this works. Okay, the first thing we're going to do is number the carbon atoms in both rings. You always want to do this because you want to make sure that you are keeping track of your atoms properly. So that's going to be the first one, and then I'm going to label the second one in a different color. Okay, great. All right, so now I want to draw the product of the condensation reaction between the following monosaccharides. Okay, so the first thing that's going to happen is this oxygen here. Let's do this in purple. This oxygen here and the hydrogen, this OH group, is going to leave, and this hydrogen is going to leave. Okay, so I'm going to specifically do the 1-4 bond. This problem doesn't specify it, so I'm just choosing this one. Okay, so here, what this means is that when I remove the OH and this H, I make H2O. So now this means that this oxygen can go ahead and bond to this anomeric carbon. Okay. But it's going to do so in the downward direction because that is where my OH group was previously. So I'm going to go ahead and draw in my new disaccharide. So this is my first monosaccharide. I'm going to go ahead and draw in all of the OH groups that are not participating in the bond. Okay, I'm going to leave the hydrogens out for now for clarity. And then that means that I still have my alpha orientation. The bond is still pointing down. And now the oxygen of my second monosaccharide is here. And then that will also be below the ring of the second monosaccharide, which is why I am pointing it down. So this bond here needs to be down on the second ring because this oxygen was down or below the ring in this structure. Okay, so this means that I have a CH2OH group up here, and then I will keep the other OH groups in their orientation. Okay, great. So what this means is that I made a 1-4, this is carbon number 4, I made a 1-4. This bond here is my glycosidic bond. This is an alpha bond. Okay. So that means that I need to classify this bond as an alpha glycosidic linkage, and it is specifically a 1-4 linkage. So I will call this 1,4 alpha glycosidic, I'm going to call it a bond, glycosidic bond, but bond and linkage mean the same thing. Okay? So that is how you would do this problem. And then I have provided the answer here in the Haworth projection so you can see it neatly written. And again, here it's identifying that this is your glycosidic bond here. It is pointing below the ring, so it is alpha. And if you number your rings in your product, I'm going to number both of them, you're going to see that that bond is between the 1 and the 4 carbon. So that's why you get a 1 to 4 alpha glycosidic bond. All right, now we are going to start with a disaccharide rather than forming a disaccharide from 2 monosaccharides. Now we're just going to start with a disaccharide and try to name the bond. So we're going to locate the glycosidic linkage in the following. All right, the first thing you want to do is locate your anomeric carbons in all of your monosaccharides. Here we only have 2 monosaccharides. So I'm going to go ahead and number the first one in green. 1, 2, 3, 4, 5, and 6. Again, this carbon here gets the 1 because it's my anomeric carbon. I know it's my anomeric carbon because if I look at my first monosaccharide, I locate the oxygen in the ring, and the carbon to the right of that oxygen is the anomeric carbon. That one is going to receive a number of 1, and then you go counterclockwise. Now look at your second monosaccharide. Locate the anomeric carbon. That anomeric carbon is going to be to the right of the oxygen in your ring, so this is going to be the anomeric for my second. So I'm going to do that in blue. This is the anomeric carbon of my second monosaccharide, so I'm going to number this. All right, now the next thing to do is locate the glycosidic bond. The glycosidic bond is going to be the one between the anomeric carbon in my first monosaccharide and then the oxygen of the second monosaccharide. So that's going to be this one right here. So this is the bond. Okay. So that means that the orientation of that bond is the one that's going to go in the name. So because that bond is pointing up in the first monosaccharide, so again, this bond belongs to this here. So you look at it relative to the first monosaccharide. Since it is pointing up, this is going to be a beta. So this is a beta bond. And now I need to specify what carbons it is bonded to. So it's going to be bonded to the first carbon in this monosaccharide, and then this oxygen, which belongs to the second monosaccharide, that one is bonded to the fourth carbon. So this becomes a 1,4 beta. So this is a 1,4 beta glycosidic bond. I will note here that sometimes you will see this notation drawn differently, so there isn't a very strict way to draw this or to write this types of bond names. Sometimes you might see it as 1,4 beta glycosidic bond. So you will see it a few different ways, but don't let that throw you off. At the end of the day, what they are specifying are the number, the carbons that are involved in the bond, and whether it's pointed up as in beta or down as in alpha. Okay. So that is how you can identify where your glycosidic bond is and how you can communicate where that bond is located in your disaccharide. So now let's go in the reverse direction. So if you can make that bond, you could probably break that bond. So if you made that bond by dehydration, so removing water, you can break that bond by adding water. And that is what hydrolysis reactions are. So we did condensation reactions or dehydration reactions where we removed water from the two from the reaction, and now we're going to add it in order to break that bond. So again, you're going to locate your glycosidic bond, which is this one here. It starts from the anomeric carbon of your first monosaccharide and then bonded to the oxygen of the second. Okay, so that means that that is the bond that we're going to break. We're going to break this bond. And when we do that, the anomeric carbon is going to get the OH, and then the oxygen that is left behind on your second monosaccharide, that's going to get the hydrogen. And that is where this H2O is coming from. Okay, so that is where it's going to go. All right, notice that you need to maintain the orientation of those new OH groups as dictated by the bond. So here for this anomeric carbon, the bond was down. So that means that in your new monosaccharide, that new OH group that you're adding to the anomeric carbon has to point down. If we look at where this bond is pointing in the second monosaccharide, we see that the bond is below the ring or down. So that means you have to maintain the OH group below the ring in that second monosaccharide. Okay. Let's go ahead and apply that. We're going to draw the products formed when the following disaccharide is hydrolyzed with water. So this means that we need to locate the glycosidic bond, and we're going to break it. And then we're going to add water across that bond. Okay, so the key to solving this is first identify your glycosidic bond. Identify the anomeric carbon in your first monosaccharide. That's going to be this one here. And then you can identify your anomeric in the second monosaccharide. That is just good practice so that you are properly oriented in what is bonding. All right, so here's the second. Okay, so this means that my glycosidic bond is going to be this one here. Okay, so this is glycosidic bond. All right, so that is the bond I will want to break. So I'm going to use this other color here, this blue, and we're going to break this bond. So what this means is that to this anomeric carbon, I'm going to add OH. And then to this oxygen, which now belongs to the second ring, I'm going to add an H. So when I draw this out, this means, I'm going to do it in red. So when I draw this out, I'm going to first draw my ring of my first monosaccharide. Go ahead and place all your OH groups where they belong. And again, you can draw Haworth projections without drawing in the hydrogens. They are implied if they're not drawn in. So this means that I'm going to have an OH group pointed down here in this anomeric carbon because the glycosidic bond initially was pointing down. So I need to maintain that orientation. That means that this is going to be an alpha anomer. Okay. Now the other, so that is going to be, that's the first monosaccharide here. Now I'm going to draw the second one, and I'm going to notice that in carbon number 4 here, the OH group has to point up in that second monosaccharide. So I'm going to, start the same process. Draw your ring first, and then I'm going to keep the orientation of all of the other OH groups, and then make sure that the OH group that was involved in my bond is in the proper orientation as well. And that is it. These would be the two. This is also an alpha anomer. Okay. But the important part here is that we maintained the proper orientation of the groups that were involved in the glycosidic bond. Okay. So those are going to be the two monosaccharides that you form when you hydrolyze this disaccharide. And here is that reaction, kind of just showing you the steps as to where everything is coming from. Okay, so that is the formation and the hydrolysis reactions of disaccharides. You just replace the OH on your anomeric carbon in the first monosaccharide, and you replace that with the oxygen of a hydroxyl group of your second monosaccharide. Okay, so now let's look at some applications of this knowledge. Let's look a little more closely into what lactose intolerance really is and artificial sweeteners. Okay, so lactose, the structure of lactose is being shown here. Lactose is a disaccharide because it has one monosaccharide and another. You're going to notice that one of the monosaccharides is galactose and the other one is glucose. So disaccharides don't need to have the same monosaccharides bonded together. You can mix and match what kind of monosaccharides are going to bond together. And lactose is an example of that. It's the principal disaccharide in milk that we consume from cows and from breast milk. So lactose consists of one galactose ring and one glucose ring, and they are joined by this type of glycosidic bond. So when we look at its structure, we can locate its anomeric carbon here, and we see it's pointing up, so we know it's going to be beta. And then if we number these rings, starting with 1 at the anomeric carbon, and then going counterclockwise. So I'm going to count 1 at the anomeric of the second monosaccharide. If we do that, we see that the bond here is between the 1 and the 4 of the second monosaccharide. So it gets the name 1,4-beta glycosidic bond. Okay. Now, where does lactose intolerance come from? Well, in order to digest lactose, we have to hydrolyze this disaccharide into its constituent parts, into its monosaccharides. And this is done in the body by an enzyme, which we're going to learn about in the section of proteins. It's done by an enzyme called lactase. Okay? And this is the one that's responsible for cleaving this glycosidic bond here. Now, individuals that are intolerant to lactose, they no longer produce this enzyme, meaning they are no longer able to hydrolyze this disaccharide. so they are not able to properly digest it, since that is a requirement in being able to digest it, is to hydrolyze this bone. So because they cannot hydrolyze it, they cannot properly digest it, and therefore they will get abdominal cramping and diarrhea. Let's look at artificial sweeteners. So first here I'm showing you sucrose, which is a naturally occurring sugar. This is our table sugar. Sometimes you see it refined as in the white variety, sometimes in the brown, but independent of how it is refined, the molecules are the same. You still have a disaccharide, which we call sucrose. And here we're going to have one that is a 6-membered ring and the other one is a 5-membered ring. So one monosaccharide is glucose, the other monosaccharide is fructose. Together, they make up sucrose, which is just sugar. Right, so now let's look at artificial sweeteners. What do these look like? Well, here I'm showing you Splenda, which is actually a derivative of sucrose. And what do I mean by derivative? What I mean is that a lot of the basic structure is the same. It's just some groups have been replaced by something else. So here we can see that if we look at the glucose monosaccharide, we see that a chlorine has been added to carbon number 4. So if we look at carbon number 4 in glucose, we see that the OH group has been removed in sucralose, which is Splenda. That OH group has been removed and a Cl has been added above the ring. If we look at the fructose monosaccharide, we see that there have been a few substitutions as well. This OH has been replaced with Cl, as well as this OH group here has also been replaced with Cl. Everything else remains the same. So this is what we mean by a derivative, right? When something is a derivative, it means there have been just minor changes made to it, but the basic structure is the same. If we compare sucralose with aspartame and saccharin, we see that aspartame and saccharin are not derivatives of sucrose because they don't have the same chemical structure. So here, this is actually going to be a derivative of amino acids, which we are going to learn about in the proteins section of the course. And this is just going to be another small molecule that, again, it's not a derivative of anything. It's just we recognize it as sweet tasting, and that is why it's an artificial sweetener. Now, why are they artificial sweeteners? Well, it turns out that these artificial sweeteners, we don't really absorb them very well because they're not naturally occurring. And so the body doesn't recognize them, even though we can taste them as sweet. And the unique thing about artificial sweeteners is that it requires only a very small amount of each compound to get the same level of sweetness that we would get from table sugar or sucrose. Okay? So what this means is that even if we were able to absorb these readily, this means that we only need a very small amount to have that same level of sweetness. And therefore, the amount of calories that we're consuming is going to be lower when we use one of these chemicals. So if we look at the sweetness between all of these molecules, you're going to see here that if we have sucrose, which is our table sugar, that is our benchmark at 1. You can see that the substituent monosaccharides, glucose and fructose is much sweeter, glucose is less sweet, and so when you combine them together, sucrose is somewhere in between. But if we look at the artificial sweeteners here, like aspartame, saccharin, and sucralose, we can see that sucralose is even 600 times sweeter than table sugar. So this means we need just a very small amount of Splenda in order to get that same level of sweetness. So that means that we can reduce calories if we consume these types of artificial sweeteners. In the next video, we are going to talk about polysaccharides, meaning now instead of having just 2 monosaccharides bonded together, we are going to have 3 or more. And so we are going to talk about the structure and function of those polysaccharides.